So chapter 22 deals with the immune system and our body's defense against pathogens. So we should start by talking about the major categories of infectious agents. First we have bacteria. Bacteria are single-celled organisms. Most of them tend to be extracellular, but it is possible to get some intracellular bacteria. So most of the time they're extracellular, but sometimes they can be intracellular. And so they're going to disrupt cell function, release enzymes that can be proteolytic, bad for our cells, but they can cause a variety of different types of disorders. So an example of a bacteria that we've probably all encountered would be the Streptococcus bacteria. You get strep throat, then you've had a bacterial infection. The next major category of an infectious agent would be a virus. Viruses are what are called obligate intracellular parasites, and that means that they are obligated to live in your cells. They cannot reproduce without getting into your cell, taking over your host machinery, and using your DNA. So these are always intracellular pathogens. Next we have fungi. And these are organisms that release proteolytic enzymes that break down your proteins and things. So athlete's foot is a fungus. So our fungi are damaging because they release proteolytic enzymes. We've got a big category called protozoans. Protozoans are eukaryotic cells that have no cell walls. They can also be intracellular or extracellular. Malaria is an example of a protozoan. And then the last major category of infectious agent is a multicellular parasite. So these are extracellular, but they reside within a host. Not a major category of infectious agent, but just something to be aware of that can infect nervous tissue. There are these things called prions that are small fragments of infectious protein that can get into nervous tissue. So just as kind of a review of the different types of leukocytes we have, we'll talk about their specific jobs in just a minute. But we met these back in chapter 18. We said we had two categories. The granulocytes that have specific staining granules. These are our neutrophils, our basophils, and our eosinophils. And then our agranulocytes lack specific staining granules. These are our macrophages or our monocytes in the blood, and our lymphocytes. We've got natural killer cells that are lymphocytes. These are involved in innate immunity. And then we've got B cells or B lymphocytes and T cells or T lymphocytes, and these are all involved in specific immunity. So we're going to talk about the specifics of each of our cell types as we go through this chapter, but just be aware that these are the cells that are helping to carry out our different types of immune responses. Find immune cells throughout the body. You find low levels of our different cells circulating in the blood, but these will be in low levels unless you're really sick and engaged in an active immune response. Then you'll find higher levels in the blood, but on a day-to-day basis they're in pretty low levels in the blood because their job is out in lymphatic tissue. So you'll find them in your lymph nodes. Your spleen houses large amounts of our white blood cells. Your tonsils as well. Tonsils are kind of like mini lymph nodes. We've got some resident macrophages that live in specific organs. The lungs have what are called alveolar macrophages. Our epithelial tissue has resident dendritic cells. Those are also phagocytic cells. Mast cells are these cells cells that are like basophils, but are found in our connective tissue throughout the body. So our connective tissue, some of them have very, very low blood supply. So if something happens and we need to get an immune response going, we need to have some cells there that are ready to deal with it. So mast cells hang out in the connective tissues and can start an inflammatory response if something is going on in those tissues. So those are our primary locations of different types of cells throughout the body. Alright, so there are these chemicals called cytokines. Cytokines, cyto always means cell, and our kines are chemicals. And these are different types of chemical messengers that aid in some type of immune response. I'm not going to make you worry about all of the specific types of cytokines that we have in the body, but we are going to talk about our interleukins today, so those are kind of important. So the first class are these things called interleukins, abbreviated as ILs. There are a bunch of different types of interleukins, but I'm not going to make you worry again about the specifics, but they are going to be important in activating the immune response, so we are going to talk about them. Other large categories are other things called TNFs, or tumor necrosis factors. We're not really going to talk about those much at all. We've talked a little bit about colony stimulating factors. When we talked about the blood, we said that if we needed to develop a certain type of cell, it would be colony stimulating factors that would call for specific types of cells to be made. So that's about all we're going to say about them now. The last major class of cytokine is what we call an interferon, and we're going to talk about these as well. And these are proteins that are released by infected cells to make other types of cells or neighboring cells aware that there's an infection and upregulate their like antiviral properties. And then interferons can also help with the inflammatory response. So we'll say they're proteins that signal other cells and can help with inflammatory response. Good on this one? Okay. So as far as our immune system goes, we have 3 lines of defense that are carried out by 2 different immune systems. The innate immune system, which provides general immunity to a wide range of things. So it's not specific. So it's general protection against a wide variety of pathogens. Then we've got an adaptive immune system, which is very specific. It involves specific T or B lymphocytes that are formed in response to encounter with a specific pathogen. So this is specific immunity due to encounter with a pathogen. So you come in contact with some disease-causing agent and we'll make T cells and B cells that are specific to that, and that is going to be what our adaptive immune system is in charge of. probably just get through the innate immune system today. We've got a lot of time before the next test, so we might start with the adaptive immune system, but I'll see how you're feeling because there's a lot of information, so I don't want to overwhelm you. All right, so our innate immune system has our first and second lines of defense. And if those both fail, then we'll engage our third line of defense, which is adaptive immunity. So let's look at each of our lines of defense. We'll talk just generally right now about what each is, and then we'll go into the specifics. So our first line of defense is going to be physical barriers that prevent entry of pathogens. So our skin is the first barrier, and then if something gets through the skin, we've got mucosal membranes, and both of those are providing physical barriers to prevent the entry of a pathogen. Something gets past your skin and your mucous membranes, and we have nonspecific internal defenses. So what are our nonspecific internal defenses? We've got different cells that are part of the innate immune response, that like our macrophages that are just going to gobble up anything foreign. We've got chemicals like our interferons or these other proteins called complement proteins that are going to act nonspecifically. Well, then we have overall physiologic responses like a fever or the inflammatory response. If both our first and second line of defense, so this is our first, this is our second, if both of these fail, then we'll kick in our third line of defense, which is the specific immunity, and we have two types of adaptive immunity. T lymphocytes, which are moderating cell-mediated immunity, so these are fighting intracellular pathogens. And then if we've got an extracellular pathogen, the B lymphocytes are going to do it. We call this humoral immunity because it's moderated through the body's humors. So your humors are your fluids, and what's happening is our B lymphocytes are going to differentiate into these cells called plasma cells, and plasma cells release antibodies to the blood. So these release antibodies to the blood. So humoral immunity is carried out in the blood. All right, so let's look at our lines of defense then. The first line of defense is going to be our physical barriers that starts with the skin. So the skin acts as a physical barrier. It's got those keratinocytes that have keratin in them, which gives some waterproofing, so that's going to help to make sure that we aren't just letting things passively in and out. So our keratinocytes contain that keratin protein that strengthens the barrier, preventing just the passive passage of fluids to and from the body. So this is going to help strengthen the barrier. Other ways that the skin is protective is that it's continually shed. So if you have gross bacteria on your skin, then it'll be continually shed off. We actually have normal bacteria that live with us. We call them our normal flora. So if we've got a bunch of okay bacteria living on our skin, then I can prevent bad bacteria, hopefully, from getting on there and replicating. So our normal flora is basically non-pathogenic bacteria to us. So it's non-pathogenic bacteria living on your skin. So it can compete for resources and hopefully compete out bad bacteria. Other things that can be produced in your skin, like sweat. Sweat contains urea and uric acid, which are both waste products for us, but it makes it hard for things to grow. So urea and uric acid can help. It acts as like an antimicrobial factor. In thick skin, Elidin helps by giving us that extra layer. Those are the ways skin is helping. Mucous membranes then are going to be our next barrier. And inside our mucous membranes, these are found in our epithelial and connective tissues. And our mucous membranes also have normal flora, which will compete out bad bacteria. And mucus itself can entrap bacteria or entrap pathogens. And then different parts of the systems are going to be able to do different things with this mucus that has pathogens trapped in it. So our respiratory tract, for example, has in our nasal cavity, we're secreting a lot of mucus. And as you breathe, those nasal concha are creating turbulence on the air that's going to whack that mucus, whack whatever you're breathing into that mucus, so that if you just inspired something, it gets trapped in that mucus, and then we can swallow it back to our pharynx. And our pharynx has a dual function in the respiratory and digestive systems. If we swallow it back in our pharynx, then it goes to our gut where we've got hydrochloric acid, and almost nothing can make it past hydrochloric acid. So we'll say that the mucus traps debris, or we'll say foreign particles, and we will say this goes to the pharynx. From there we can swallow it. It'll make it to your stomach. Your stomach has hydrochloric acid. And almost nothing can beat the hydrochloric acids. So that'll dissolve a bunch of stuff. So that's going to be the first part of our respiratory tract that's involved with providing immunity. But there's also this thing further down in your lower respiratory tract called the mucus escalator. And we've got all of these ciliated cells. Cilia move mucus along the surface of a cell. So these ciliated cells are beating mucus up the respiratory tract so that, again, we can get it to the pharynx and swallow it. So that's another part of the respiratory tract that's going to help. It's called the mucus escalator. And it's ciliated cells in the lower respiratory tract that beat the mucus up to the pharynx. You good on this? Okay. All right, so in our GI tract, we have several things that are going to help provide some immunity. Saliva has some antimicrobial enzymes. The stomach has hydrochloric acid. So that acid can break up and kill, hopefully, anything that we ingest. Our colon has another huge colony of normal flora, or your large intestine, also known as your colon, has what we call our gut flora. So this is a huge colony of bacteria that lives in your intestine. It processes all your undigested material and really kind of helps to protect against bad bacteria. And then then in our digestive tract or in our gastrointestinal tract, we can also induce vomiting or diarrhea. So both of those would be innate ways to get rid of pathogens. So vomiting and diarrhea are also ways to rid the system of pathogens. We good? Okay, the next in the urogenital tract, this is where we are producing urine. Urine is also high in urea and uric acid, and it's sterile, so it's going to be able to help to clean out your urogenital tract. And then also throughout the urogenital tract, we've got a weak acid, lactic acid. But if it's acidic, it makes it hard for microbes to live in it. So those are going to be our external barriers, or the barriers that we have to entry into the system. So that's our very first line of defense. Any questions? So this is nonspecific. It's providing protection against a wide variety of pathogens. But if it fails, then we have to get to our second line of defense. And this is going to be carried out by specific types of leukocytes. So, well, first, so our cells are going to be the first thing that is going to help in our second line of defense. But we also have antimicrobial proteins that are going to be able to activate or help in our second line of defense. So first, if we look at what our neutrophils and macrophages are doing, these are actively phagocytic cells. They can both do this thing called the respiratory burst. They do it different ways. Neutrophils can do respiratory burst on their own. Macrophages can be stimulated by T helper cells. T helper cells or T sub H, we'll come back to those guys in a minute. So this other thing that our neutrophils and macrophages can do is called respiratory burst. And basically what they're doing is they're releasing oxidative chemicals. That's how it gets the name respiratory. They are releasing oxidative chemicals that kill everything in the area. So macrophages are being stimulated to do it. Neutrophils do it on their own. How does that happen? Our neutrophils hanging out here, these guys primarily eat bacteria. So they fight bacteria. And let's say this neutrophil just realizes, wow, I can't gobble up any more bacteria. There's so much bacteria in this area. Then what it can do is it can take all of its lysosomes. Lysosomes are those organelles inside the cell that have digestive enzymes, and it sticks them up towards its surface, and they degranulate. So our neutrophils put all lysosomes at the plasma membrane. And they degranulate, it's called. What does this mean? They release all of their digestive enzymes. They digest the neutrophil, so it kills itself. But in killing itself, then it releases all these digestive enzymes that can kill any bacteria in the area. So the major component of pus is neutrophils that have undergone respiratory burst. So they've degranulated, They've released all their digestive enzymes and they're killing all the bacteria all around them. They call it a kill zone because everything right around that nothing right around it can survive. They degranulate. So that's just like they let go of all of their enzymes, it ruptures their cell wall, but then it creates this kill zone all around them so that nothing there can live. Macrophages you should primarily think of as being phagocytic. And another thing that macrophages are going to do is they're going to become antigen-presenting cells, or APCs. This is going to be important for activating our specific immune response. So that's another big job of our macrophages, is to become antigen-presenting cells. Are we good on this? Okay. Then our next cells are basophils and mast cells. Mast cells are basophils living in connective tissue. So these are like basophils, but they're residents in connective tissue. So if you think about like our cartilages, our tendons, our ligaments, they're not highly vascularized. So if you have damage there, we need to have some cells that can start an inflammatory response. So basophils are going to be involved in the inflammatory response, which is a normal response to tissue damage. And what they do is they release chemicals that are going to attract other immune cells to the area, and they release other chemicals, histamine and heparin, that are going to act as vasodilators and anticoagulants. So they attract other immune cells, and they release histamine, which is a vasodilator, and they release heparin, which is an anticoagulant. So that in an inflammatory response, we can increase our tissue permeability, get these other immune cells to the system. We don't want to start clotting right away because we want to bring these other blood cells to the site of tissue damage so that we can begin the inflammatory response, get in contact with any bacteria that may be in that damaged tissue, and bring healing factors also to the area. So we'll talk more about the inflammatory response in a little bit. Any questions on basophils or mast cells? All right, the next leukocytes that are involved in innate immunity are natural killer cells. So these are a type of lymphocyte, but they're not specific. These are going to induce apoptosis in cancer cells or in virus-infected cells. So these induce cell suicide, which is called apoptosis, in virus or cancer-infected cells. So what these cells do is they will encounter a cell that's sick. So let's say this is a sick cell. A natural killer cell will come along, and it's going to it's going to punch a hole in the membrane with these things called perforins. So it uses perforins to punch a hole in the membrane. And then it injects a lethal dose of this cytotoxic chemical called granzymes. A lethal dose of granzymes. And the granzymes are going to cause this cell basically to shrivel up and die. So the cell will shrivel up and die. And our last type of innate cell are eosinophils, and these are going to do a couple things. They release cytotoxins that paralyze parasitic worms, and that's what they do as part of our second line of defense. They have other functions in cleaning up, so I wouldn't necessarily consider it a second line of defense, but we should still talk about their other functions. They gobble up or phagocytize antibody or antigen-antibody complexes. And they can also gobble up some allergens. That's not really a part of the second line of defense. That's more of a cleanup mechanism for after something else is going on in the body. But we should just be aware that's another thing our eosinophils do. All right, questions on our cells of innate immunity. You good? All right, so then the next thing that is going to provide some innate immunity are what are called antimicrobial proteins. Interferons we talked about a little bit ago, but what interferons are these paracrine chemicals that are released by infected cells that will cause other cells in the area to upregulate their antiviral properties. They can also attract natural killer cells, and they can also attract macrophages. So interferons are going to be able to do a couple of things. So let's say we've got a virus-infected cell. We can say that they're released by virus-infected cells and signal all neighboring cells to upregulate their antiviral proteins. Another thing that interferons can do is activate natural killer cells so that they can induce apoptosis in viral infected cells. And then the other thing they're going to do is attract macrophages. So that they can gobble up our infected cells. Our next group of antimicrobial proteins are called the complement system. This is like around 20 proteins that all act in the immune system to do different things. So it's about 20 different complement proteins. And they are going to help to mark pathogens for removal from the system through very specific mechanisms. So first, what's called opsonization. And in opsonization, you are basically marking the pathogen for removal by a phagocyte. So when the complement protein binds it, now the macrophage is going to recognize that this needs to be gobbled up. So we'll say mark for removal by a macrophage. So opsonization is the first thing our complement system is going to do. The second thing it's going to do is it's going to stimulate inflammation. We'll talk about the inflammatory response in just a bit. The third thing our complement proteins are going to do is what's called cytolysis. So cyto, always referring to cell, lysis is the destruction of a cell. So what they do, if this is our like a bacterial cell or yeah, mostly like a bacterial cell or some other cellular pathogen, then what they can do is you'll get a bunch of complement proteins line up and form what's called a membrane attack complex, or a MAC. And so you form these little membrane attack complexes all over the cell, and then whatever's inside the cell can get out, whatever's outside the cell can get in. Basically, it just lyses the cell. So our membrane attack complexes cause cell lysis. Then the last thing our complement proteins are going to do is they'll be able to mark something for elimination from the immune system or elimination from the system. So how this can happen, like it'll help if we've got an antibody that is bound to a pathogen, then one portion of the portion that's not bound to the antibody can bind to a complement protein, and then it can be taken out of the system. We can stick it to an erythrocyte and filter it out in the spleen. So we'll say elimination of immune complexes, and this can be used to clean antigen-antibody complexes out of the system. So those are the four things complement proteins do: stimulate opsonization, which is marking it for removal by a macrophage; stimulate the inflammatory response, we'll talk about in a little bit; stimulate cytolysis by forming these membrane attack complexes, which will cause the cell to lyse; and then they'll help with immune clearing. Some books call this immune clearance. So it's the elimination of our antigen-antibody complexes.
Do they also, what is it, it's agglutination?
Those are antibodies.
Okay.
Yep. I mean, I guess as part of this, you could kind of think of part of this elimination as doing that. You're sticking it to a red blood cell and then pulling it out. But the antibodies are going to be more, we're going to consider doing that. All right, next, inflammation. Inflammation is a general response to any type of tissue damage. So, what are the functions of inflammation? So, inflammation is going to do several things for the body. It's going to help to mobilize our defenses. By our damaged tissue will secrete these chemicals that are going to attract our immune cells. So, we'll attract immune cells to site of injury. Going to be the first important function. So our damaged cells will release cytokines that are going to call other immune cells to the area. These chemicals are also going to increase the tissue permeability, so those immune cells can get through the tissue and or through the blood out to the tissue to do their fighting. The next thing it's going to do is contain the pathogen, so we'll localize it to one area. So fibrinogen will leave the blood and form a fibrin network around the damaged tissue so that we can contain whatever pathogens might be in it. So fibrinogen will be pulled from blood. And will form a fibrin mesh around the area to localize pathogens. That way they can't spread too far. And then the last function of inflammation is going to be to help with tissue cleanup and repair. So 8 to 12 hours after the inflammatory response has happened, we have our monocytes there, or they've become macrophages. We've got neutrophils there, cleaning up the area, and then platelets will start healing the tissue. So neutrophils and macrophages are eating up debris. So these are taking care of debris, bacteria, dead or damaged cells. So our neutrophils and macrophages are taking care of all of that cleanup part, and then platelets will begin our tissue repair. Good? All right, so what are the cardinal signs of an inflammatory response? Redness. This is caused by increased blood. So due to increased blood to the area. Heat, which is also due to increased blood. Swelling, which is a result of the increased blood, but also if we're increasing blood flow to the area, then we're increasing extracellular fluid. We're going to call that exudate, and that's going to cause swelling in the area. So we'll say an increase in exudate is going to cause swelling, and then as we swell more, we get more and more tissue fluid. So this is interstitial fluid. And we'll talk about the specifics about how we get more to the area in just a minute. This increased exudate, as we swell in the area, we're going to activate pain receptors. So pain is the last cardinal sign of an inflammatory response, and this is due to activation of nociceptors in response to swelling. All right, so how do we get an inflammatory response going? What's happening? So the first thing that's going to happen is our damaged tissue is going to release what are called chemotactic factors. So these are chemicals that are going to do several things in the cell. So damaged tissue releases, they're also called, in general, they're cytokines, but these chemotactic factors, chemo is referring to chemical, tactic is like movement. So we are using chemicals to cause movement of immune cells to the area. So it releases, we could also just say this is in the general group of cytokines that are going to do a couple things. They're going to increase tissue permeability so that we're going to be able to get more cells through to the tissue so that we'll get more blood flow to the area. But this, we'll say these chemotactic factors are going to, will attract more blood cells to the area. And stimulate our vascular changes, which is the second step. We'll talk about the specific vascular changes. So the second step in our inflammatory response are these vascular changes, which is a result from all of those chemicals that are being released by the damaged tissue. So what's going to happen is, as far as vascular changes go, we'll have increased permeability due to we are going to dilate the blood vessels in the area, and then we're going to open up the intracellular cleft between them. So we'll dilate arterioles, and we will increase permeability by the cells kind of loosen up. They've got intercellular clefts in them, and those are going to kind of open up wider so that the capillaries become more permeable. By, let's say, kind of increasing the size of the intercellular clefts in the capillaries. And then the other thing that's going to happen is our capillary cells, our endothelial cells, are going to express these things called cell adhesion molecules, or CAMs. So, endothelial cells express CAMs. CAMs are cell adhesion molecules. And if we look at what that looks like, our little endothelial cells, those intracellular clefts are going to get bigger, and they're going to start sticking up these little CAMs, these proteins that they can stick up on their surface that are going to slow down the immune cells that are going to come in our next step. So as more of these chemical factors are out there in the system, then our white blood cells will be drawn from the blood. This blood is rushing past very quickly. So we've got to slow the immune cells down and let them get out. So our third step is the recruitment of our immune cells. And this is going to happen as our neutrophils and macrophages respond to those chemotactic factors. So they're the first to arrive. Neutrophils come first, shortly followed by macrophages. So we'll say they are attracted to the area. When they roll along those cell adhesion molecules, it slows them down. That's called margination. So the cells roll along the endothelium, sticking to those CAMs. The next thing they do is what's called diapedesis. So if we look at those cells again, if this is and they're displaying their CAMs, and the blood is flowing by very quickly, our neutrophils and macrophages are going to start slowing down and rolling along the wall. That's this margination step. The next thing they're going to do is called diapedesis. Dia always means through, pedesis, this is like movement. And so what will happen is they'll squeeze between these intercellular clefts and squeeze out to the tissues. So the cells squeeze out of capillary and into the tissues. And then they will also start releasing chemotactic factors to attract more cells to the area. We good on that? Okay, so then the last part of the immune or the inflammatory response is delivery of plasma proteins, and we've got increased blood flow to the area, and so as we get increased amounts of blood to the area, more plasma is going to be squeezed out. So we'll deliver plasma proteins that contain antibodies that can bind up bacteria if we've met it before. If we haven't, we're going to have to start a new immune response. But that's going to be one part of it. And then this, as kind of this delivery of all this extra blood, we're going to increase the interstitial fluid. And in this case, we're going to call it exudate. And that's going to be important because all of that extra interstitial fluid, where do you think it's getting picked up? Who's picking it up? Yeah, the lymphatic capillaries. And then we can take that lymph right past the lymph node and expose whatever pathogens may be in it to specific immune cells. So we'll increase the interstitial fluid. This becomes what we call exudate. The exudate gets picked up in lymphatic capillaries. It'll go to our larger lymphatic vessels. Those vessels are going to run past the lymph node. And in that lymph node, we can start a specific immune response. So that's a really important part of the inflammatory response, is increasing all of that fluid in the tissues so that if there was any foreign object in it, we can pick it up in that exudate, get it to a lymphatic capillary, which is ultimately going to drain it into a lymphatic vessel that's going to run past the lymph node, and we can start a specific immune response. So fever is an abnormally high body temperature that's maintained due to the presence of some pathogen. And it's going to be important for the body for a couple reasons. So this is abnormally high body temperature. It's caused because cells like macrophages and cells that are infected will start releasing these chemicals called pyrogens. So when your hypothalamus senses pyrogens, it knows that we can let your body temperature go up. I'm not going to test you on the specifics of that. What I do want you to be aware of is what are the protective functions of fever. So if we have a high body temperature, we can increase metabolism. And what this is going to do is it's going to help with tissue repair. Another thing fever does is it actually stimulates your spleen and your liver to sequester iron and zinc. Sequester means hold it all up for yourself. And microbes need iron and zinc to function. So if we hold up all of our iron and zinc, then the microbes can't get it. So our liver and spleen will sequester iron and zinc. And we'll say that these are needed by microbes. So if we hold them up, then the microbes can't have them. And then the other thing that the fever should do is mess with the enzymes and things in the pathogen, so it can interfere with their ability to replicate. So it can hamper or dampen microbial replication. The other thing that it's going to do is it's going to increase interferon activity. So low-grade fever is actually good to have for a while. So you don't want to have a high fever for very long at all, because then you're going to start actually messing with your own enzymes and damage your brain. But a low-grade fever can be good. And so when your kids get fevers a lot because kids are meeting a ton of pathogens for the first time, so they're getting sick. And they may be uncomfortable, but it can actually be good to help, like, wrap them up in a blanket even though they're hot and make them stay hot for a little bit, because it's going to help their immune system. So I took an immunology class, and my teacher always said that allowing all of these things to go on for a little bit is good. So an inflammatory response for a little bit is good because it's helping to get that exudate to the area. It's helping to get that lymph then to the lymph nodes. a fever for a little bit is good because it's helping to increase our metabolism. It's really helping to hamper the replication of those microbes, increasing our interferon activity. That's going to be really important for a little bit. A sinus infection where you're making a ton of mucus is actually good for a little bit because then you can swallow that mucus and digest those pathogens that are replicating in your nose. So none of it's good for a long, long time. Eventually you do want to do some symptom management. Like if you're going 5 days of still snot in your sinus infection, it's not doing anything anymore. Your body is in the process of mounting an immune response, a specific immune response, so you can start taking your Sudafed or whatever. If your child is really, they're in a lot of pain and they want to take a fever reducer, then okay, give them the fever reducer, but then bundle them up so that their body temperature is still a little bit higher. So all of these things do have important roles in the body. Our first tendency is to want to just take something to feel better, but they're there for a reason. They're helping us to mount an immune response in some way. So that's all of our innate immunity.